Okay, I would like to thank STEMCELL Technologies for allowing to share our work in this webinar. And I would like to tell you about our work on human enteroendocrine cells using intestinal organoids. And if there's a bit of time left, I also like to share a few slides on our corona work. So most people, if you think of the gut, you think of nutrient absorption or cancer, but it's actually also home to most endocrine cells of any mammal organ. There are at least 6 different types of these endocrine cells, which we call enteroendocrine cells. These are organized based on their main hormone product that they secrete. There are about 20 different hormones in the gut, and these hormones regulate different kind of things related to the nutrient response, such as insulin secretion. They regulate your appetite, gut motility, and they do this by signaling to distant endocrine organs, but also to the central nervous system. So because of this important function in relation to the nutrient response, they are very good targets for metabolic diseases. One good example of a hormone that's already targeted is GLP-1. So this is a hormone that induces insulin secretion, and it's used for treating diabetes. So the most important class of treatments that were used here are the DPP-4 inhibitors. DPP-4 is an enzyme that degrades GLP-1. If you inhibit this enzyme, you have more GLP-1 and you stimulate insulin secretion. There's a molecule called exendin-4, which is a molecule that looks like GLP-1 but cannot be degraded by DPP-4. This is isolated from the Gila monster, a reptile, from its venom. We're actually culturing the venom gland organoids from this guy now and see if we can find similar such molecules. And now there are also new GLP-1 receptor agonists, which seem very powerful for type 2 diabetes treatments. But there are many other hormones that are not effectively targeted yet. So one of the challenges with enteroendocrine cells is that although the gut is very big and there are many endocrine cells, it's still only less than 1% of the cell number. There are also different hormones between humans and mice. For example, humans have motilin. It's a very important hormone in humans that regulates, for example, gut motility, but that mice don't have. Also the sensory receptors, so the receptors that enteroendocrine cells use to sense nutrients are different between mice and humans. So currently there are no good physiological systems to study these features which are unique to human enteroendocrine cells. We think there are 2 very important outstanding questions. One is, how do enteroendocrine cells decide which hormone they produce? So as I said, there are about 20 different hormones. And what are the triggers for secretion of these molecules? So we would like to use organoids to assess these questions and see if we can use those to identify new therapeutic targets. So we can induce enteroendocrine cells reasonably well with small molecules, playing around with signaling factors. But the most efficient way in human organoids is actually an overexpression of a transcription factor that's called neurogenin-3. It's the transcription factor that drives the generation of all endocrine cells in the gut. So what we do, we have a doxycycline-induced overexpression pulse of neurogenin-3. And we do this in different parts of the gut, different organoids from different regions of the gut, because all these different regions have different hormones. We see that these differences are maintained in the organoid cultures. So, for example, duodenal organoids will have hormones that you find mostly in the duodenum. An ileal organoid, distal small intestine, will have hormones that you normally find mostly in the distal small intestine. So if you look at these organoids by immunofluorescence, we see what we call normal co-expression patterns. That means that we see hormones that we never expect together in one cell, we never see together. For example, here serotonin, GLP-1, and somatostatin, we never see together in one organoid. So these enteroendocrine cells seem normal, even though they are generated by overexpression of a transcription factor. So morphologically, looking at transmission, electron microscopy, they look normal. And the most functional hallmark of an enteroendocrine cell is secreting a hormone, obviously, and they do that very well. So even though we can make organoids with about 70% being enteroendocrine cells, some of these hormones are still relatively rare. So we would like to have fluorescent reporters to follow these hormones and also purify them in high amounts. So we can do that by using HDR. We use CRISPR-Cas9 cutting at the stop codon of a gene and using HDR to knock in a fluorescent protein. This is not very efficient. It's also taking a lot of time to make all these factors. So a strategy that we now mostly use is based on crisp paint, a strategy that uses null homologous end joining to cut the stop codon as close as possible to the stop codon, and we just provide a piece of DNA coding for the fluorescent protein that will just be built in. So how do we do this? We electroporate a mixture of cells with plasmids coding for a guide RNA targeting the stop codon of the hormone locus, and we provide the fluorescent protein that's to be integrated. We electroporate that, we sort the transfected cells, we clonally grow them out, and then the resulting organoids, for some hormones we see 1% correct insertion, for some hormones we even have 20% correct insertion. And we find those by giving these organoids a brief pulse of neurogenin 3 overexpression, doxycycline, and wherever we see appearance of tagged hormones, we will pick these organoids and clonally expand them. So using this strategy, we already built about a few dozen of hormone reporters. So here are a few examples. You see here the fluorescent reporter, and here the stain, the immunofluorescent staining for the same hormone. You always see perfect overlap. And we can also do this as multiple times after each other to make multiple hormone reporters. We already have some organoids with 3 different hormones being tagged. And here you see one example. So one thing that we've learned in mice about enteroendocrine cells is that there are important differences between the crypt and the villus. So some hormones are only made in the crypt, like GLP-1, and some hormones are enriched in the villus. So using lineage tracing, we found that these crypt enteroendocrine cells can indeed in the villus change the hormone they make. This is a mouse model here to the right, where we could look in time at the expression of different hormones in individuals over time. And they indeed see that some hormones go up and then go down, after which others go up. So it seems that these enteroendocrine cells can change over the crypt-villus axis. So using organoids, in this study in 2018, we looked at which factors actually regulate this hormone switching. And we found that BMP signaling, which we normally keep low in organoid cultures, if you activate that, you get expression of villus-enriched hormones, like secretin here, and you repress crypt-enriched hormones. So BMP signaling is normally low in the crypt and high in the villus. So that's actually pattern consistent with these hormones being changed. So if we go to mice and inhibit BMP signaling, we could indeed, these hormones are normally restricted to the crypt, would out of a sudden also be expressed in the villus here. So they would change their crypt restriction to also being expressed in the villus. Villus-enriched hormones such as secretin would decrease. So we're interested if a similar kind of regulation would also be happening in human enteroendocrine cells. Here the example of this GLP-1, which is actually coded by the messenger glucagon, is also enriched in the crypt, just like in mice. And indeed, in our organoid cultures, where we overexpress neurogenin 3, if you give these organoids BMP signals, then you could completely repress GLP-1 and induce this neurotensin, NTS, which is a villus hormone, here quantified on mRNA to the right. Somatostatin, SST, which is a hormone both found in the crypt and villus, doesn't change. Here, using our reporter organoids at the bottom, we could indeed see that BMP signals would prevent the appearance of GLP-1 positive cells, but also reduce the levels of GLP-1 in existing GLP-1 positive cells. So it seems to be exactly doing the same as in mice. So next we used the system to figure out if we could see other kind of signaling pathways that would regulate hormone expression. So we modulated activities of known players in the intestinal crypt, such as BMP, Wnt, and Notch signals. And we did this not only after the induction of neurogenin 3, but we also did it before the induction of neurogenin 3. And that is because we see that neurogenin 3 can be induced at different positions in the crypt axis. So not only here just outside the stem cell compartment, but it could also be induced higher in the crypt, which means that these cells will be exposed to different levels of different signaling pathways, these different morphogens. So one striking thing that we observed is, as we know before, we see that BMP activation after neurogenin-free expression represses glucagon or GLP-1, which we know. But we also see that Wnt inhibition before neurogenin-free overexpression represses glucagon, but induces motilin. And you see that here to the bottom in immunofluorescence, we get a great reduction in GLP-1 with this pre-Wnt inhibition and a huge overexpression of motilin. So it seems to be that even the state of a cell when it starts expressing neurogenin-free could influence the eventual fate of the cell. So next, we use our organoids to build a deep atlas, both transcriptomic and proteomic atlas of all the different enteroendocrine cell subtypes. So using our reporters, we could build both a bulk enteroendocrine cell subtype RNA atlas and a proteome atlas. And here you see the single cell atlas. We have over 2,000 enteroendocrine cells from humans. We did this both in the presence and absence of BMP signals to create both these crypt and villa states of enteroendocrine cells. And one surprising thing that we seen, again, we could see that these L cells, which are the cells that make GLP-1, if you activate BMP, so these are violin plots showing the transcript counts in single cells. So if you would activate BMP signaling, you would again get a repression of this GLP-1, and you would also get an induction of neurotensin, exactly as we've seen before. But we also see changes in other hormones. And this is the hormone pair ghrelin-motilin. These are made by the same cell. We call them MX cells. And we see that BMP signals repress ghrelin and induce motilin. And this is surprising. As said, motilin doesn't exist in mice. So this is a change we have not seen in mice before. But this could be another crypt-villus hormone pair that is dynamic over this axis, regulated by BMP signals. To the right, you see BMP target genes that act as a validation to show that we have proper BMP activation. So next, we try to use our data sets to mine for new receptors, new transcription factors, new hormones that regulate enteroendocrine cells. And how we do this is using different kind of reporters, in this case, a calcium reporter, to follow activation of the enteroendocrine cell in real time. So most enteroendocrine cells use GPCRs, G protein coupled receptors, to sense luminal components of the gut, and in response to that, secrete their hormones. And these GPCRs can be coupled to a calcium response. So what we do, we put calcium, encoded calcium sensors in our multicolor organoids to follow calcium responses in individual enteroendocrine cell subtypes. For this, we actually used the first blue calcium reporter developed by Joachim Goedhart in the University of Amsterdam, because we by now are using up our colors in the organoids. And this example shows an olfactory receptor, which is highly expressed in a subset of enteroendocrine cells. That is actually sensing this molecule called beta-ionone, or aromatic flavor from this plant. And if you add it to organoids, you can indeed see calcium activation in a subset of enteroendocrine cells, and here quantified. So we can use these organoids to measure enteroendocrine cell subtype-specific responses to stimulants. And we predict that this actually would be coupled to a secretion of a hormone that induces satiety. Another surprising thing is that we know that enteroendocrine cell subtypes can express receptors for other hormones from enteroendocrine cells, so that there's communication between enteroendocrine cells. This is very well known for the hormone somatostatin, but we also see a lot of expression of two other hormone receptors, basically for secretin and PYY. So here the secretin receptor is shown, expressed in these populations that express GLP-1. And this is the mouse expression of the secretin receptor. So mouse enteroendocrine cells basically don't express it at all. Here, confirmation on the human intestinal slide, you indeed see secretin receptor RNA being co-localized with this enteroendocrine cell marker. So if you add secretin to the organoid medium, you actually can see swelling, which is related to activation of GPCRs that signals through cyclic AMP. which activates CFTR and induces swelling. That's the chloride channel. And this is generally something you also see with forskolin. And if we look at the supernatant of organoids and measure GLP-1 in this case, because that's the hormone where the secretin receptor is expressed in, we can see induction of GLP-1 release. So showing that this receptor that we find in these GLP-1 producing cells seems to be regulating hormone secretion. One of the enteroendocrine cells where we see by far the most differences with the mouse counterparts are these cells that make ghrelin. So as said, the human cells that make ghrelin also express motilin, this hormone that mice don't have. And probably because of that, there are many differences associated with that in these cells. For example, we see expression of angiotensin, a very well-known blood pressure regulator. We see this IL-20 receptor that we also could confirm in vivo being expressed. And we see this molecule called cerebellin-1, which actually has been shown to be an appetite suppressant, just like ghrelin. So actually should say appetite inducer. So that's consistent with each other. And we're now looking into this IL-20 receptor seems to actually be able to regulate the secretion of motilin. But yeah, as I said, especially these cells show a lot of differences with the mouse counterparts. Transcription factors. So these different subtypes of enteroendocrine cells have a very specific set of transcription factors that seem to be important for their generation. Also there we see a few differences with the mouse counterparts. For example, ALCL1, which is a transcription factor very well known from the lung neuroendocrine cells, seems to be highly expressed in many human enteroendocrine cells, but not in mouse. And what we do with this is we test the function of these transcription factors in human enteroendocrine cells by knocking them out using CRISPR-Cas9. And here are two examples. HHEX, that's a transcription factor expressed in this population called the delta cells that make somatostatin. If you knock this transcription factor out, you lose the expression of somatostatin. And here, quantified using immunofluorescence, you see a complete loss of these delta cells. LMH1A, another example, expressed in the cells that make serotonin, here marked by chromogranin A and TPH1. TPH1 is the enzyme that is rate limiting in the synthesis of serotonin. If you knock out LMH1A, you see a strong reduction in this enzyme. Surprisingly, we didn't really see a decrease in serotonin. This could mean that maybe TPH1 in vitro is not rate limiting in the synthesis of serotonin. And it also means that LMH1A is at least not important for the generation of these cells, but maybe more for their functioning. So these enriched organoids, enriched for enteroendocrine cells, give us the unique opportunity to start looking at their the products that they secrete. Because they're so pure, there are barely any other cell types. We can just take the supernatant. The supernatant basically corresponds to the basolateral side of the cells, because the inside of organoids are apical, the outside is the basolateral side. And this is also why the hormones would be secreted to. So we can just take the supernatant from these organoids and perform mass spec analysis on them. We've done that with Baiwu and Julia from the Utrecht University, and have started looking into what these cells secrete. So we compared the secretome with the intracellular proteome of enteroendocrine cells. We found that these secretome proteins are heavily enriched for GO terms associated with secretion. So giving confidence that indeed we're looking at secreted proteins. And here you can see ranked lists of the protein abundances that we found in the supernatants. And indeed, the most abundant proteins in the supernatant are always hormones. So the top 100 are basically mostly hormones. Here, there are a few annotated. And if we look at the control organoids, we mostly found factors that are coming from goblet cells, where we barely see hormones being high up in the list. So giving extra confidence that we're actually looking at the secreted products from human enteroendocrine cells. So to the left here, a heat map of known hormones that we can find. Basically, they are ranked in the total secreted protein and processed peptides. So these are the fractions smaller than 10 kilodalton, and these are the fractions larger than 10 kilodaltons. We have separated them. And these are generally processed peptides. Enteroendocrine cells hormones are basically produced as a big pre-pro-hormone, and then are proteolytically cleaved in many steps to generate the eventual bioactive peptides. So basically we separate these two to distinguish these two fractions. And using this analysis, we could also see the some secreted products that we have never seen in mouse enteroendocrine cells. We could also find in the human supernatant. We predicted these already from our RNA analysis, but with this analysis, we could also see that they're actually secreted. So giving confidence that these could be hormones or secreted products that affect other hormones, such as some enzymes that we actually also see here that mice don't have. To the right, we look at, these are the peptides that we actually measure in the mass spec, and these are different kind of bioactive peptides. As I said, these pre-hormones are cleaved into different kind of peptides that are bioactive, and we can look at, do we see all of those, or do we see differences between different regions of the gut? So there's another kind of thing we can do with this data. So one thing we are currently doing is some high throughput screens using our fluorescent reporters. So based on our transcriptomic data, we can do some biased analysis of potential EC regulators. For example, we can see a receptor in our RNA analysis, and we can stimulate it and see secretion of a hormone. But sometimes we don't know what is the ligand of a receptor. We also think that we miss many receptors because they are lowly expressed. Then we see many transcription factors unique to enteroendocrine cell subtypes, but we don't know how these transcription factors are often regulated. So what we try to do is using our fluorescent reporters to perform high throughput screens where we do unbiased screens. So for example, we just throw GPCR agonists on the organoids and see what happens. So we actually see that our fluorescent reporters, because these hormones are linked to the fluorescent protein, they're coupled, it's a fusion protein. We can actually see secretion of the fluorescent protein. So we hope that this can be used in such a way to have an easy readout of secretion of a hormone. Secondly, we also use these fluorescent reporters just as a readout of production of the hormone. So we are thinking of developmental screens where we could skew the enteroendocrine cell production into one hormone instead of another, and thereby provide or generate a favorable response therapeutically, for example, for diabetes treatment. So just to summarize this enteroendocrine cell part, we've molecularly characterized the human enteroendocrine cells, and we think we build an accessible platform to study their functioning using the fluorescent reporters in combination with the neurogenic-free overexpression. One thing that we still are going into, and I haven't been talking about today, is the microbiota. There are many kind of bacteria in the gut that are related or correlated to protection against obesity or type 2 diabetes, but how this really works is not known. So we're combining these bacteria with enteroendocrine cells to see what is their effect on the gut hormones. And another project that we're continuing on is the regional differences in the hormones along the gut. So as said, different parts of the gut make different hormones, but how these differences are really regulated is not known. We know that these differences are stable in organoid culture, so they will persist forever, so that they must be some way hardwired, for example, through DNA methylation. So we're currently looking into which kind of regulations this could be doing that. So a little sidestep to SARS. So based on the reports of very high ACE2 expression in the human gut, ACE2 is the receptor for the SARS-2 coronavirus. Very high expression in the human gut, especially in enterocytes, based on RNA studies. And also in this picture of the Human Protein Atlas that is confirmed. And some patients of COVID-19 have some gastrointestinal symptoms, and there's also some viral RNA measured in part of the patients in the stool. So we thought to use intestinal organoids to see if we can use it as an infection model of SARS-CoV-2. So we've combined intestinal organoids with the coronavirus, with SARS-CoV-2, and this is a collaboration with the Erasmus University in Rotterdam. And we found that intestinal organoids actually very efficiently harbor infection and can propagate the virus. So at short-term infection, 24 hours, we often see only a single cell infected organoid, not more than that. But only 2 days later, often these organoids completely turn into a full infected organoid, with most cells being infected. So clearly indicating that this virus can very efficiently spread inside the intestinal cells in the organoids. But surprisingly, we see that proliferating cells are infected very efficiently as well. So we see here marked by Ka67 proliferating cells, and here in white double-stranded RNA marking the virus. Most of the cells are proliferating in these organoids, and these are all infected. We also see infection of enterocyte, here stained by apolipoprotein A1, which is expected. But this proliferating cell infection is a bit unexpected because they barely express ACE2. So looking at RNA-seq of different conditions where we have either expanding organoids or differentiated organoids, we can indeed see that these expanding organoids barely express ACE2, and upon differentiation it goes up about 1,000-fold. Yet these differentiated organoids, they infect as efficiently as expanding organoids, and expanding organoids generate live viruses over a time course, maybe even more efficiently than differentiated organoids. We found that the addition of BMP induces even higher ACE2 levels in enterocytes, but even that doesn't really make the infection more efficient. So we think that either ACE2 is acting is enough at very low levels to propagate infection. It could also be that the intestine has maybe alternative receptors that could facilitate entry of the coronavirus into the cells. And we're looking into that now to see if there are any of such receptors. We try to see if those exist in the gut. So with that, I would like to end. Thank particularly Jens, whom I've been doing all this work with on the human enteroendocrine cells, and Adriana, who was our student and is now continuing her PhD work in the lab. And with that, I would like to end